Imagine your body is running on outdated software, a single line of code that is glitching, causing a disease that no pill can fix. For decades, we could only manage the symptoms of these glitches. But what if we could send in a microscopic team to find that broken line, delete it, and replace it with a perfect version? We are no longer just practicing medicine. We are performing a biological software update. This is the power of gene therapy, and it is the ultimate expression of the Research Codon. Welcome back to my channel. I'm the Geneticist. Today in Research Codon, we are exploring the bridge between theory and cure. We've talked about embryos and digital twins, but today we look at the patients living among us whose lives are being saved by rewritten DNA. This is how we modify the very essence of a cell to fight back against destiny. Gene therapy isn't just one technique, it's a toolkit. Sometimes we use gene addition, where we deliver a healthy copy of a gene to compensate for a missing or broken one, a method that has already changed the lives of those with hemophilia. Other times, we use gene editing, like CRISPR, to physically cut and correct a mutation. And for some conditions, we use gene silencing, using molecular mutes like RNA interference to shut down harmful genes before they can cause damage. We are learning to play the genome like an instrument. But how do you get a new gene into 1 trillion living cells? We use the most efficient delivery experts on the planet, viruses. Scientists take viruses like AAV or lentivirus, gut them so they can't make you sick, and pack them with therapeutic DNA. These modified viruses act as biological delivery trucks, sneaking into the cell and dropping off the repair kit exactly where it's needed. It is a masterpiece of bioengineering, using nature's own invaders to become nature's healers. Perhaps the most incredible application is cell-based gene therapy. In treatments like CAR-T, we actually remove a patient's own T cells, the soldiers of the immune system, and genetically engineer them in a lab. We give them a new radar that allows them to recognize and destroy cancer cells. Once these super soldiers are put back into the patient, they hunt down the disease with a precision no chemotherapy could ever match. We are turning the patient's own body into the ultimate drug. While viruses are effective, they aren't perfect. They can trigger immune responses or be difficult to scale. This is why the Research Codon is moving toward non-viral methods. We are developing lipid nanoparticles, tiny fatty bubbles, and direct injection techniques that can deliver genetic material with lower risk. As these delivery systems become more refined, gene therapy will move from rare miracle treatments to a standard part of modern healthcare. The goal is simple: to make incurable a word of the past. We are living through the era where DNA has become a medicine. We are the architects of a future where your genetic destiny is no longer set in stone. If you want to explore the clinical success stories and the molecular pathways of the viral vectors I discussed today, you can find the full research on my Telegram channel. Thank you for joining me for this episode of Research Codon. Stay curious, keep those cells dividing, and I'll see you in the next decode. See you soon.